Hello everyone, today we will explore the blood physiology with special focusing on the RBCs or the red blood cells. We will first review the general function and the basic compositions of the blood. Let's begin with the function of the blood. In a simple way to remember them, it is transport, defense, hemostasis or sealing, and stabilization. First, the blood acts as a transport system. It carries the oxygen from the lung to the tissues and carbon dioxide back from tissue to the lung, and of course transport the nutrition and the hormones. Second, blood is an important defense because it contains the immune and the antibody. Third, the blood is essential for homeostasis and hemostasis. First, the hemostasis, which means stop of the bleeding, and this is achieved by the platelet and the clotting factor in the blood. Finally, the blood contributes to stabilization of the internal environment or the homeostasis. It maintains the pH, fluid balance, and temperatures of the body. Now, let's move to the compositions of the blood. The blood is divided into two main components, the plasma, which make 55%, and the cells, which represent 45%. For the plasma, we can find a water mainly, and gases, inorganic substance, and organic substance. The common example for inorganic substance is sodium chloride. The organic substance is the proteins, lipids, and the glucose. For the cell, we have three main types of the cells: the red blood cells or erythrocyte, white blood cells, leukocyte, and finally the platelet. Here we are going mainly to discuss two important things. First, the plasma protein, then the main topic for today is the blood cells. So let's start with the plasma. Let's move to the plasma protein and their function. A simple effective way to remember is ABCDTV. A stands for the albumin. Albumin is the main protein responsible for maintaining the oncotic or osmotic pressures in the blood because it's a large molecule. It cannot leave the capillary, so it retains water. Second, B, buffering. Plasma protein contribute to the buffering of the and maintaining of the blood pH, in addition to the bicarb system. C stands for clotting and capillary function. Regarding the clotting, plasma proteins such as the fibrinogen prothrombin play a central role in hemostasis, help stop the bleed, as we are going to discuss in the coming videos. And second, the capillary function. Plasma proteins, especially the albumin, regulate the capillary permeability and fluid exchange. The defense, D. Some plasma protein, mainly the globulin, they are function as an antibody, so they are essential for the immune response and the protecting of the body against pathogen. T, the transport. The albumin acts as a carrier protein transporting the hormone, drugs, and other substances. V, the viscosities. Plasma protein, especially the fibrinogen, contribute mainly to the blood viscosity. So, the blood viscosity affects the blood flow, making this factor, which is fibrinogen, one of the important factors that controlling the blood flow. So, here please remember that the function of blood, transport, defense, hemostasis, and homeostasis, while the plasma protein function is ABCDTV, albumin for osmosis, blood buffering, capillary and clotting function, defense, transport, and the viscosities. Now let's begin with the RBCs or the erythrocyte. Important to know that the RBC is called corpuscle rather than true cells because it lacks nucleus most of organelle. And in this part, we are going to cover the function, the shapes and structure, the erythropoiesis or the production of RBCs, and the fate lifespan of the RBCs. First, starting with the function of the RBCs. The main rule of RBCs is summarized in 3K function. The hemoglobin carrier, the RBCs act as a container for the hemoglobin, prevent the hemoglobin from excreted through the kidney, and it also responsible for the gas transport. The RBC is essential for transporting their main respiratory gases, the oxygen carried by hemoglobin. As for carbon dioxide, there is a carbonic anhydrase enzyme which convert the carbon dioxide into bicarb. Then the buffering hemoglobin also functions as an important buffer system that maintaining the blood pH with normal range. What about the shape and the structure of the RBCs? The RBCs is biconcave. This biconcave provides two major advantages. First, increase the surface area, and this enhance the gas exchange and also allow the cell to expand when the water enter, reduce the risk of damage. Second, the great flexibility allows the RBCs to pass easily through the narrow capillary without rupturing. Now let's focusing on the structure. The RBCs mainly is a corpuscle with no nucleus, with no organelle, mainly hemoglobin, cytoplasm, and some enzymes. Therefore, the RBCs has a limited lifespan, approximately 120 days, after which they are removed from the circulation. Finally, the hemoglobin concentration. The hemoglobin inside the RBCs is about 34 grams per 100 milliliter of the cell. This value cannot be exceeded due to the limited metabolic capacity of RBCs to sensitize the hemoglobin. Now, let's move to another point about the RBCs. Now let's move to the next key concept, the erythropoiesis or the production of the RBCs. In this section, we'll cover three main points: the site of production, the step of formation, and the key factor responsible for the formation of the RBCs. First, the site of production. The site of erythropoiesis changes by age. So, with early embryo, the RBCs formed in the yolk sac. With the mid-gestation, the production shifts to the liver, spleen, and lymph node. After birth, the bone marrow becomes the main permanent site for RBCs production. What about the step of erythropoiesis? The RBCs develop through the well-organized sequence, starting with pluripotent stem cells, which is converted to the colony-forming unit erythrocyte, then erythroblast, then the reticulocyte, and the mature RBCs. Please remember, as the cell becomes more mature, the nucleus gradually disappears, the organelle lost, and the cell becomes filled with the hemoglobin. This transformation allows the RBCs to be specialized in oxygen transport. So, also remember that the stem cell, erythroblast, and reticulocyte present in the bone marrow, but reticulocyte need to move from the bone marrow to the blood, and in the blood the reticulocyte mature to RBCs. So remember that in the blood we can find the reticulocyte 1% only and the RBCs. Also remember that the account or the number of reticulocytes is important indicator of the bone marrow activity. So if we have anemia and a low reticulocyte number, this means that the cause mainly in the bone marrow. If we have a normal or a high reticulocyte and anemia, so the bone marrow is function well and the cause is another, not the bone marrow. Let's discuss the essential factor regulating the erythropoiesis, and we will focus on three key factors: the erythropoietin, the vitamin B12 and folic, and the iron. First, start with the erythropoietin, which is the primary stimuli, the trigger that give the order to bone marrow to produce the RBCs. Where is it produced? 90% of the erythropoietin, which is a hormone by the way, is formed in the kidney and 10% in the liver. Here I want to emphasize that kidney failure decreases the erythropoietin, decreasing the RBC, so patients with renal failure normally they have anemia. What triggers the erythropoietin release? The main stimuli is the hypoxia, and hypoxia increase the hypoxic inducible factor that stimulates the kidney and liver to produce more erythropoietin. So what are the causes of hypoxia? First, the high altitude with a low atmospheric oxygen, lung disease, low oxygen entry, heart disease, low oxygen pump, reduced blood flow, low oxygen transport, anemia, low oxygen carrying capacity. It is important to note that either we are talking about the high altitude, lung, heart, or blood flow as a contributor or cause for the hypoxia. All these factors will increase the erythropoietin, increasing the number of RBCs, so they may increase the viscosity, unlike the anemia in which the erythropoietin increase to restore the number of the RBCs back to the normal. So, second factor is the vitamin B12 and folic acid, and both are essential for the DNA synthesis, which is critical for cell division and maturation. So when they are deficient, when we have a lack of both of them, we have an impairment in cell division, and this results in a large RBCs, which is called megablast, with low number of RBCs, which is anemia. Together it is a megaplastic anemia. That's to say big RBCs, but they are low in number. And also the cells, the RBCs become more fragile, so easily destroyed. And now let's move to the core part, or the core factor, or the most important factor is the iron. It's the core for the hemoglobin formation, therefore the oxygen transport. So before this, we need to review briefly the hemoglobin structure. The hemoglobin consists of two main components: the heme, which is derived from the succinyl-CoA Krebs cycle, converted to pyrrole, and we need 4 pyrrole to form a protoporphin, and we need protoporphin to combine with the iron to form the heme. Second part is the globin, which is a polypeptide made of amino acid, and we have 4 types: alpha, beta, gamma, and delta. Hemoglobin formation starts with a heme bind to the globin, forming one hemoglobin chain, and we need 4 chains to form one hemoglobin molecule. We have 2 types of the hemoglobin: the adult hemoglobin, a hemoglobin A, which is formed of 2 alpha, 2 beta, and fetal hemoglobin, which is formed of 2 alpha and 2 gamma. Fetal hemoglobin have the higher affinity for oxygen, which is essential for the fetal life. So the type of the polypeptide is essential since it determines the affinity to oxygen. Another important point that the oxygen is bind mainly to the iron in the hemoglobin. So let's move to the iron metabolism. What are the sources of the iron? The diet is the main sources of the iron. The meat, which contain the hemoglobin and myoglobin. The iron is absorbed in small intestine and is transported by transferrin. So iron bind to apotransferrin to form transferrin in the blood. We cannot see an iron alone in the blood. Then the utilization of the iron, the transferrin delivered the iron either to bone marrow to be used in formation of the hemoglobin, or to the muscle to form the myoglobin, and the excess iron is going to be stored in the liver in the form of the ferritin, which is the main storage form of the iron. So iron bind with the apoferritin to form the ferritin. However, if we have excess iron with less ferritin, or less apoferritin, so the iron is stored in another form, which is a hemosiderin. So it is important to note that the iron in a ferrous form, Fe2, important to bind the oxygen, and it is used for the formation of the hemoglobin, while the ferric cannot bind to the oxygen, and we cannot use it for the formation of the RBCs. So first, iron transport by transferrin, stored in the liver, or go to the bone marrow to enter in the process of the erythropoiesis. Let's talk about the final point in our topic today. So now let's move to the final topic, the fate of the RBCs. Here we will focus on the lifespan and the breakdown recycling. For the lifespan, the red blood cells can have an average lifespan of about 120. After this period, they become less flexible and removed from the circulation because first the enzymes in the RBCs synthesis start to decrease, and the ATP, which is responsible for the transport and maintain of the membrane integrity, also decreased. So that's why they die after 120 days. Where they are destroyed mainly in the spleen and in the liver, and both organs have a macrophage which recognize and remove the old damaged RBCs. So once the RBCs are destroyed, the hemoglobin released and taken by the macrophage. Inside the cell, the hemoglobin broke down into main component, the globin, which broke down to amino acid, and these amino acids reused for synthesis of new protein, polypeptides, and globin, and the heme, which is further divided into the iron and protoporphyrin. The iron recycled in our body, stored first in the liver. If it's needed, it will be transported by transferrin to the bone marrow or the muscles, and the protoporphyrin, which is converted to the bilirubin, and the bilirubin is excreted in the bile. So after 120 days, the RBC is destroyed, the hemoglobin released, the hemoglobin divided to the globin, reused, the amino acid, the iron recycled or restored, the protoporphyrin convert to bilirubin and excreted in the bile. So let's recap. We have now reached to the end of today's topic. Let's briefly recap the key points in clear, organized way. First, the blood function. We talked about the blood in general, function and composition. We started with the main function of the blood, which can be summarized into transport, defense, hemostasis, and homeostasis. Then we discussed the composition of the RBC, the blood, which is plasma and the cell element. Then we moved to the plasma protein, highlighted the function of plasma protein, which is ABCDTV, albumin, buffering, clotting and capillary defense, and transport and viscosities. Remember, the blood function differ from the plasma protein functions. Then we move to the red blood cells or erythrocyte, and we discuss the function that they carry the hemoglobin, transport oxygen and carbon dioxide, and act as a buffer. Structure, they are biconcave, increasing the surface and flexibility with no nuclear or organelle, and the hemoglobin content is limited by the metabolic capacity. The production or erythropoiesis, the site is embryo, in the yolk sac, liver, spleen, lymph node, but after birth mainly and only the bone marrow, the step from the stem, proerythroblast, erythroblast, reticulocytes, and RBCs. And as we move from the proerythroblast to the RBCs, we lose the nucleus, organelle, and replaced by the hemoglobin. The essential factor is erythropoietin, mainly from the kidney and to less extent from the liver, and stimulated by the hypoxia. And it is the signal for the bone marrow, or it gives an order to the bone marrow to produce the RBCs. Then we talked about the vitamin B12 and folic acid required for DNA synthesis. So when they are deficient, the RBCs become large and fewer, and they become also fragile. The iron is essential for hemoglobin formation. So we reviewed the hemoglobin structures, which has two main parts: the heme, which is protoporphyrin and iron, and globin, which are polypeptide chain alpha, beta, gamma, and delta. For the iron metabolism, we discussed the absorption, small intestine, the transport by transferrin, storage in the liver, mainly by ferritin or hemosiderin. The fate of the RBCs after 120 days, the RBCs destroyed in spleen and liver. The breakdown results in the globin, which is recycled, iron recycled, protoporphyrin converted to bilirubin, excreted in the bile. So that's all for today. See you in next episode.